Hello, Mr. Richard.
Hello, Miss Christine. It's nice to meet you.
Nice to meet you, sir, as well. It's my pleasure that you are here with us on our podcast today despite your hectic schedule. It's an honor, sir.
The pleasure is all mine.
Sir, we will start quickly as you have several meetings today as well. Sir, the whole podcast is going to be based on your journey. We want to know how it all began and how you became the Richard Ebright.
Yeah, I grew up as only child in my family in north of Reading, Pennsylvania. My hometown, but there was nothing much I could do much, but there was one thing that kept me interested.
What was it, sir?
Collect things.
So he did, and did he ever? Beginning from kindergarten, Ebright collected butterflies with the same determination that has kept him going at the age as well. That will that doesn't let him down. He also collected rocks, fossils and many more things throughout his career so far. He is more than one personality. At times he becomes an eager astronomer as well. He had a driving curiosity along with a bright mind.
My mother also played a key role and always encouraged my interest in learning. She took me on trips, bought me telescopes, microscopes, cameras and other equipment that kept me motivated and kept alive the eagerness to research.
I was his only companion until he started school. After that I would invite home friends for him, but I kept the night time for him in my schedule so we can enjoy together. Richie has been my lifeline since he was in 3rd grade, when his father passed away. I used to spend every evening with him at the dining room table. If he didn't find things to do, I used to assign him some work, like got him involved in learning things. Richie was always eager to learn. And so he did. Richard was a very bright student, who earned top grades in school. On everyday things, he was just like every other child. By the time Richard arrived in 2nd grade, he had collected all 25 species of butterflies found around his hometown. He caught monarchs, whites and brush-footed butterflies and many more.
That would have been the end of my butterfly collection, but then, my mother got me a children's book.
What was the book?
The Travels of Monarch X. That book told me that how monarchs used to migrate to Central America. That opened the world of science to this young collector.
But how did it?
At the end of the book, readers were invited to help study butterfly migrations. They were asked to tag butterflies for research by Dr. Frederick Urquhart of the University of Toronto, Canada. After reading that book, I soon began to tag butterflies. Anyone who found a tagged butterfly was asked to send the tag to Dr. Urquhart.
The butterfly collecting season lasts around 6 weeks in cat summer in Reading. Ebright's next step to raise a flock of butterflies. He told that he used to catch a female monarch, take her eggs and raise them in his basement through their life cycle, from an egg to a caterpillar, to a pupa, to an adult butterfly. At last he used to tag the butterfly's wings and let them go. For years, his basement was home to thousands of monarchs within their developmental stages.
Eventually, I began to lose interest in tagging butterflies.
But why?
It was so tedious at that time. In all due time, only 2 butterflies I had tagged were recaptured. That too, they were caught between 75 miles from me.
Richard got his first taste of real science when he was in 7th grade. He then understood what science was. That too when he entered a county science fair. And he lost.
It was really a sad feeling to sit there and not get anything while everyone else had won something or the other.
But you were such a brilliant student. Why did you lost? And what did you prepare for the fair? Can you brief a little about it?
My entry was slides of frog tissues, which I showed under a microscope. The reason I lost was that the winners had tried to do real experiments, not simply a neat display.
Your sadness till today shows how determined you are and you were.
I knew that for the next year's fair, I would have to do a real experiment. And the subject I knew most about was the insect species.
So what was your next step?
I wrote to Dr. Urquhart for ideas, and you know him. He came back with a stack of suggestions for experiments.
So what was next?
Nothing more. Those ideas kept me busy all through high school and led to several prize-winning projects in county as well as international science fairs.
Mr. Richard, Sorry for interrupting, but can you brief a little about the projects you did?
Yeah, for sure. Like in my 8th grade, I tried to find the cause of a viral disease that kills nearly all monarch caterpillars every few years. I thought that the disease might be carried by a beetle. So then, I tried to raise the caterpillars in the presence of beetles. But I didn't get any results. Still, I showed that I had tried such an experiment, and fortunately this time I won the contest.
What were your further discoveries? As far as I have heard you so far, I know you wouldn't stop there. Am I right?
You are correct with your assumption. The next year my project was to test the theory that tells that the viceroy butterflies copy the monarchs.
Yeah, I have heard that theory.
The theory was that viceroy look like monarchs, because monarchs don't taste good to birds. Viceroy, on the other hand, do taste good to birds. The more they look like monarchs, the less likely they are to become a bird's dinner.
What was your project focusing on, then?
My project was to see whether in fact birds would eat monarchs. I found out that a starling would eat a monarch rather than ordinary bird food. This time for my work, I was placed first in the zoology division and third overall in the county science fair.
That is it. You didn't stop.
I didn't. In my 2nd year of high school, I began a research that led to the discovery of an unknown insect hormone, and eventually that led to my then new theory on the life of cells.
But what were you trying to do actually?
The question I tried to answer was a simple one.
What was it?
What was the purpose of the 12 tiny gold spots on a monarch pupa?
I thought the spots were just ornamental.
Nothing to worry, that's what everyone thought. Those who didn't agree were me and Drive. Urquhart.
Ebright and another fellow excellent science student then had to build a device that showed that the spots were producing the hormone necessary for the butterfly's full development.
This fair got me first place in the county fair and entry into the International Science and Engineering Fair. There I won 3rd place for zoology. I even got a chance to work during the summer at the entomology laboratory of the Walter Reed Army Institute of Research.
Then, what was next?
As a high school junior, Richard Ebright continued his advanced experiments on the monarch pupa. That year my project won the 1st place at the International Science Fair and gave him another chance to work in the Army laboratory during the summer.
In his senior year, Richard went a step further. He grew cells from a monarch's wing and showed that the cells would divide and develop into normal butterfly wing scales, only if they were fed the hormone from the gold spots.
That project won 1st place for zoology at the International Fair. Then I spent my rest of summer working in the laboratory of U.S. Department of Agriculture. Then I got into Harvard, and then after the 1st year at Harvard, I went back to the Department of Agriculture and continued to research more on the hormone from the gold spots. There, using the sophisticated instruments of laboratory, I was able to identify their hormonal chemical structure. Then after about 1.5 years, I got an idea for my new cell theory.
How did you get this idea from?
Interesting. I got it while I was looking at X-ray photos of the chemical structure of a hormone.
But do you know that after this achievement, Ebright didn't scream or anything like that, rather he believed that. Along with his research about the insects, the photos gave him the answer to one of the puzzles of biology, that how a cell can read the blueprint of its DNA.
After completing the riddles of biology, I, along with my college roommate, James R. Wong, worked all night drawing pictures and constructing plastic models of molecules to show how it could happen. Together we wrote the theory paper as an article in Proceedings of the National Academy of Science.
And as I remember, that was the first time that journal published the work of a college student?
Yes, you remember right.
Ebright then went on graduating from Harvard with highest honors, second in his class among 1,510. Ebright then went on to become a graduate student, a researcher at Harvard Medical School. There I began to test my theory. If the theory proves correct, then it will be a big step towards understanding the processes of life and further leading to new ideas that lead to prevent some types of cancer and other diseases. What all led to this? Ebright's high school research into the purpose of spots on a monarch pupa led him to his cell theory. But did you know that Richard is not only keen towards science, but also has several other traits, like he is a champion debater, a champion public speaker, a good canoeist, and an all-rounder in general? He is also an expert photographer as well, particularly wildlife. In high school, Ebright was a student. Because learning was easy, he spent his energy on the debating and model United Nations clubs. Was there anyone you admired?
Yes, Richard Ebright, my social studies teacher and advisor. He was the perfect person for me. He opened my mind to new ideas.
Let's see what Mr. Ebright has to say about Richard. Mr. Ebright, we know that you know Mr. Richard very well. Can you share your experience with him? What made him so special?
Richard would always give that extra effort. What pleased me about him was, he was that person who put in 3 or 4 hours at night doing debate research besides doing all his research with butterflies and other interests. He was always competitive, but not in a bad sense. Richard wasn't interested in winning to win a prize, or for just the sake of winning.
Then, why do you think Richard used to win?
He won because he always wanted to do the best job he could. He wanted to be the best.
Making of a scientist includes these ingredients. Starting with a first-rate mind, with added curiosity, and mixing the will to win for right reasons. Richard had all these qualities.